Hello, everyone, and welcome to today's webinar, Diabetes and the Clinical Laboratory. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker. Professor Garry John has played a central role in establishing HbA1c diagnostic testing and guidelines worldwide. He has chaired several committees within the IFCC, including the HbA1c Working Group, who developed a standardization procedure of HbA1c measurements. He has worked closely with numerous national and international diabetes organizations and has been an advisor to the WHO. Professor Garry John will now begin the presentation.
For that kind introduction. Today I'm going to talk to you in some depth about hemoglobin A1c analysis and its clinical utility. These are my disclosures that this is a program that is supported by Abbott, although I don't represent Abbott in any way whatsoever. As some learning objectives, hopefully by the end of this lecture, we'll be able to understand the testing method principles of hemoglobin A1c. We will review with you the methodologies, and we'll discuss it as a diagnostic and monitoring tool. And I will go in some depth into quality specifications, and we just maybe have a chance to overview guidelines for diabetes testing. Measurement science and diabetes have a long relationship interlinked over many, many years and centuries. Indeed, going back to the middle of the 1600s, a British physician and biochemist called Willis described the urine collected from diabetics as having a sweet taste. Indeed, he associated this with good fellowship and guzzling down of wine. A century later, Dobson went slightly further, slightly more scientific, and he isolated a dry residue from the urine collected from diabetics, and he said it looked and tasted like brown sugar. Luckily, something we don't have to do anymore. Glucose remained the mainstay of diabetes testing for many centuries, and it wasn't until a chance finding by Rabar back in 1968 who described an abnormal hemoglobin in the blood of patients with diabetes, that this fraction came to the interest of the clinical fraternity. We now know, of course, that this is not an abnormal hemoglobin, but indeed is glycated hemoglobin or hemoglobin A1c. I don't intend to go into biochemistry in any great depth, but we need to know that this is a non-enzymatic reaction and follows typically the law of mass action. Things to remember about this reaction. It's a two-stage reaction. It goes through a relatively quick reaction, then a very slow amidal rearrangement. Overall, the process is slow and continuous. This reaction continues throughout the lifespan of a red cell. And need to remember that glucose binds preferentially to the N-terminal valine of the hemoglobin, but the glucose will bind to other sites on the hemoglobin molecule. If glucose binds to the N-terminal valine, then it creates a charge difference on the hemoglobin. If glucose binds elsewhere on the hemoglobin molecule, it does not create a charge difference. And it's often said that hemoglobin A1C represents the previous 3 to 4 months of glucose control within the individual. And that is true to a large extent. But because it's a continuous process and continual turnover, what we are seeing is that a blood sample collected on a patient, the glycated hemoglobin A1C that is measured reflects about half of the measured hemoglobin A1C is reflected within that month. So 50% of the effect of that result is what you see within that month. About 25% the previous month, and only about 6% is reflected 4 months previous. So yes, hemoglobin A1C does reflect the previous 3 to 4 months, but the major effect on the result is what you see in the previous 1 month. So, what happens when hemoglobin is glycated? As I said, if it is glycated at the N-terminal valine, it creates a slight charge difference between glycated hemoglobin and non-glycated hemoglobin. There is a structural difference. Obviously, it now has a glucose attached to the structure. And there is a chemical difference. And each of these have been used to actually devise methods for measuring hemoglobin A1C. Let's first look at charge difference. Cation exchange chromatography is probably the most widely known of the methods to measure hemoglobin A1C. It's been around for a long time, and there are many, many different manufacturers that produce instruments for measuring hemoglobin A1C using ion exchange, cation exchange chromatography. And there are numerous advantages to this method, but also several disadvantages. It was the original method described for measuring hemoglobin A1C, and it is well proven. And there are many, many manufacturers. That is a good or a bad thing. It has good separation, but be careful, that is very much manufacturer dependent. It identifies common hemoglobin variants and excludes them from analysis. Again, be careful, it is manufacturer dependent. It standardized the IFCC reference measurement procedure, something I will come back to later, but again, be careful, depend on the manufacturer. It is interfered with by a number of factors, again, depending on the manufacturer. So yes, probably the most widely known of the methods, but it has its advantages, also it has its disadvantages. Still on charge separation, we have capillary electrophoresis. This is electrophoresis is similar to ion exchange chromatography, but done in a column or in a capillary rather than the column. This again has a number of advantages. This does show excellent separation, good accuracy and precision, but it has disadvantages like there are many manufacturers that actually have variable quality. It does not have a stat ability, and one has to question whether it's clinically needed or not. And it has a slow throughput, therefore several capillaries have to use in parallel to cope with the throughput required for clinical purposes. They can go on structural difference. There's a glucose attached to the hemoglobin, and affinity chromatography makes use of this glucose. The boronic acid on the affinity column recognizes the cis diol associated with glucose and loosely binds it on the column, so that any hemoglobin that has got glucose attached to it will stick to the column, and hemoglobin without glucose just washes straight through. But the main difference here is that this recognizes hemoglobin that is glycated at any part of the hemoglobin molecule, not just the N-terminal valine. So you're not measuring hemoglobin A1C as such, but calibration will bring it back to hemoglobin A1C. This again has a number of good advantages. It shows good accuracy and precision, easy sample handling, no interference from hemoglobin variants, and no interference from non-glucose adducts. But it does have disadvantages. There is some batch-to-batch variability in gel quality. It requires a dedicated analyzer, as does HPLC and capillary electrophoresis. But it does not identify abnormal hemoglobins. And if that's important, and we can discuss this possibly in the questions, then this system will not show that abnormality up. You got immunoassay. Immunoassay is a system which detects the glucose bound to 2, 3, or 4 amino acids. It's been around for some time now, and it's quite amazing to think that you could raise an antibody to something where the only difference is the glucose moiety. Again, it has a number of advantages and disadvantages. It shows good accuracy and imprecision, but it is very analyzer dependent. The better accuracy and precision is found if it is dedicated an analyzer used to measure it, rather than on the main chemistry analyzer within the laboratory. It has easy sample handling, and because it only detects about 3 or 4 amino acids, there is no interference from hemoglobin variants, apart from the very rare amino acids, very rare variants that change amino acids at positions 1 to 3. No interference from non-glucose adducts, and can be done on routine chemistry analyzers. But as I say, that results in poorer performance than it is done on a dedicated analyzer. Again, it does not recognize hemoglobin variants. And finally, chemical differences. This is probably the most recent methodology you find now on the market, enzyme immunoassay. This uses a protease to cleave so the 2 amino acids, creating a fructose dipeptide, and then uses this enzymatic reaction to produce color. Again, this shows incredibly good accuracy and imprecision, easy sample handling, and like immunoassay, has no interference from hemoglobin variants apart from those at positions number 1 or 2. It isn't interfered from non-glucose adducts and can be done on routine chemistry analyzers, and it can have a large sample throughput. Disadvantages are very small, but possibly we are still learning how this assay works over long-term external quality assessment. It does not identify hemoglobin variants, and it also means putting whole blood onto your main analytical system. But overall, there are a number of different means of measuring hemoglobin A1C. One is not better than the other. It depends on your situation. You choose the method that best suits the laboratory that you are serving. So, getting the correct hemoglobin A1C result. Why is this so important? Well, if we look here at the data from the UK PDS, this is in type 2 diabetics, we could see that a change in only 1% hemoglobin A1C, a very small improvement in hemoglobin A1C, resulted in a 21% decrease in deaths related to diabetes, a 37% decrease in microvascular complications, and a 14% decrease in associated myocardial infarctions. So we have to have methods that can well differentiate a 1% change in hemoglobin A1C. So there was a need to standardize. And why was this? Well, if we look at the CAP surveys going back to the early 1990s, we can see that there's a huge variation in the results being obtained with different methods. Not only were the results targets being different and poor accuracy, but we're also seeing poor imprecision. If you look in the UK at the same time, things didn't look good. In the laboratory, things look even worse. This shows here between laboratory agreement for different methods used in the clinical laboratories in a large region within the UK. And you can see here that depending on which laboratory you went to, you could actually have a result in one lab of 4.3% hemoglobin A1C. Or you went to a lab down the road and you get a result of 9.4% hemoglobin A1C. There was no standardization. And when we're trying to look at differences of 1% to see these large clinical differences, you can see here that these results have little or no clinical relevance. So this lack of global standardization resulted in a number of national schemes being introduced. And probably the most widely recognized was the National Glycohemoglobin Standardization Program, now of course called NGSP. There's also the Swedish scheme and the Japanese standardization program. But the major problem of these national schemes were that there was no reference method. There was no primary reference material and no traceability. What does this mean? Well, the NGSP setup was a wonderfully conceived project. It resulted in a core primary reference laboratory running a highly optimized hemoglobin A1C method. This CPRL was then used to standardize secondary reference laboratories. And the secondary reference laboratories were then used to calibrate routine methods that we would use in the laboratory. The primary reference method being used at the time was a optimized HPLC described by Goldstein, Little et al., which was produced and published in the mid-1980s, and is now recognized as having significant positive bias due to interference. So as this does not have a reference measure procedure, this is harmonization, not standardization. What do I mean by this? Well, very simply, if we look at a dartboard and we throw in darts to the dartboard, and we're throwing them and we've got good grouping together. In other words, we get all the results to be the same. But if you're aiming for the bullseye, we get the results wrong. This is harmonization. All the results are harmonized to a given value or given standard, but it's not the true value. Whereas if you throw in the darts again, aiming for the bull, and you get the same kind of grouping, then this is standardization. You're getting the right result. But harmonization does have an effect. If you go back and look at the same CAP survey, we can see now that harmonization has resulted in those mean values, getting far closer to what you expect. In precision, it remains poor, but the target values, the accuracy is getting better. So it might not be true standardization, but harmonization has helped. But the IFCC has recognized there's a need for global standardization of hemoglobin A1C and produced a working group to look at this. And over the years, this working group has produced a standard method. And this method that we tried to produce was based on methodological requirements. And there are levels of methodological requirements we need to actually understand. We need to identify first the measurand. What are we going to measure? A single measurand is required. We need to develop reference measurement procedures that accurately, precisely measures the measurand. We have reference laboratories, reference material, and need to be traceable. Let's look at the measurand first. We decided the measurand will be hemoglobin with glucose attached to the N-terminal valine. This is called hemoglobin beta chain blood N1-deoxyfructose 1-aryl hemoglobin beta chain, or hemoglobin A1C. That is a measurand. That is a single entity, a glucose attached to the N-terminal valine. Also, it was recommended that we measure this property in SI units, and the SI unit for hemoglobin A1C is millimole per mole. Millimole of hemoglobin A1C per mole of hemoglobin A0 plus hemoglobin A1C. We need to develop a reference measurement procedure that accurately measures hemoglobin A1C and is not interfered with by any other property that may be there. And if we are to develop a reference measurement procedure, we need a standard to calibrate this reference measurement procedure. This is what we've done. We have produced pure hemoglobin A1C and pure hemoglobin A0. And these are used in the reference measurement procedure, which is based on enzymatic cleavage and then measurement following HPLC and mass spec, or HPLC and capillary electrophoresis. Both give identical results. Both are highly specific for hemoglobin A1C and not interfered with by other fractions. This method is not a method we would use in the routine laboratory. This method is only used in reference laboratories that are used to assign values to standards which we would use in a clinical laboratory. We need to identify reference laboratories. And now this reference measurement procedure is used in a large number of laboratories worldwide. Every single one of these labs runs the reference measurement procedure, and every one of these laboratories has input into assigning calibration values to standards that we would use within our clinical laboratories. Not one laboratory is used to assign value, all laboratories are, and that leads to a very high level of accuracy. So we now have a method that we know measures hemoglobin A1C specifically, is used in a large number of laboratories around the world, all of which are used to assign values to standards that we use in a clinical laboratory. And it is now traceable. We now have an unbroken chain of traceability from the patient sample that we measure in routine laboratories back to the primary reference material, which are banked at the IRMM. So we now have a stable, traceable method that we can use to assign our patient samples to. And when we look at the consensus statement produced by the American Diabetes Association, the European Association for Study of Diabetes, the International Diabetes Federation, and the IFCC, it says that the IFCC reference system, which I've just described to you, represents the only valid anchor to implement standardization of the hemoglobin A1C measurement. And now we are lucky that all major instrument manufacturers calibrate their systems using standards provided by the IFCC laboratory network. So we now have a system whereby we've got long-term stability and accuracy, so we can measure our patient samples with a high level of confidence. And now we look at standardization, we can see that standardization has resulted in an even better outcome for our standards. The mean results now lie far closer to the target values, showing that accuracy has improved. And with accuracy improving, we are finding that imprecision is also improving. The question that one or two people ask me as well is, why are HbA1c results from IFC standardization different to those from NGSP harmonization? Why is a hemoglobin A1c of 7% not the same as a hemoglobin A1c of 70 millimoles per mole? Well, that goes back to looking at the NGSP value compared to the IFCC calibration. We see here the lines of a relationship between the NGSP, the Japanese system, and the Swedish system, and they correlate well. But if we look a bit more closely at the bottom end, we see that there's a difference of about 1.5 to 2% hemoglobin A1c, with the NGSP values being higher. And that reflects the non-specific interference that we see in the NGSP reference method. And this is taken into account when we calculate the values and relate them from the NGSP to the IFCC. So yes, there's a relationship there, but because of this non-specific interference, results are not the same. Let's move on now to clinical aspects of hemoglobin A1c. From the DCCT trial of many years ago now, we could see that as hemoglobin A1c values increased, so did the rate of retinopathy, nephropathy, neuropathy, and microalbuminuria. And as you can see, the higher the hemoglobin A1c, the greater the increase in these conditions resulting from diabetes. Let's look at it the other way. If we were able to lower the hemoglobin A1c, we could decrease the levels of these conditions. As you can see from this graph, just decreasing by small amounts had remarkable improvements in patients, in this case with type 1 diabetes, but also with type 2 diabetes. So what targets should we look for? Well, the American Diabetes Association, the ADA, the European Association for Study of Diabetes, the ESD, and the International Diabetes Federation, the IDF, have all issued the recommendations, but they differ. The ADA and the ESD have target hemoglobin A1c values of 53 millimoles per mole, or 7%. But the IDF, who focus more on postprandial hyperglycemia, have a target value of 48 millimoles per mole, or 6.5%, quite different. In the UK, NICE guidelines, National Institute for Clinical Excellence, suggest that we measure hemoglobin A1c in type 2 diabetics at 3 to 6 monthly intervals, while the glycemia is changing, and possibly 6 monthly intervals once glycemia is in a stable state. And that we use hemoglobin A1C has calibrated the IFCC standardization. And we have a target of 48 millimoles per mole, 6.5% for type 2 diabetics as a whole, or if they're on drug treatment, then in case of hypoglycemia, we aim for a slightly higher value of 53 millimoles per mole, or 7%. I don't expect you to go look at this in any great detail, but we then produce very detailed guidance of what to do if values fall above this. Now, with type 1 diabetes, we suggest that hemoglobin A1C should be measured every 3 to 6 months, considered more often in adults with type 1 diabetes if the glucose control is suspected to change rapidly. And again, the method should be calibrated to the IFCC. Because of the associated long-term risks with type 1 diabetes, a target level of 48 millimoles per mole, or 6.5%, is attempted to be aimed for. But as we now know, we should agree individualized hemoglobin A1C with a target for each adult with type 1 diabetes, and this will differ from individual to individual depending on this situation. Again, worldwide, there is a huge global burden associated with diabetes. Back in 2011, there were about 366 million people with diabetes. By 2030, less than 10 years away now, this will have risen to 500 odd million. Speed of increase, this is already suggested to be surpassing this by 2030. But possibly the most worrying thing is that about half of people with diabetes globally are undiagnosed. This is a major impact on individuals, and we have to try and address this. Currently, we are treating diabetes possibly too late. This is type 2 diabetes. There's a move away from normality through dysglycemia into frank diabetes. And we're currently diagnosing patients with diabetes at about this point. At this point, they've already significantly had increase in microvascular complications, but possibly more importantly, there's been a huge increase in macrovascular complications. Ideally, we should be finding patients far earlier so we can intervene earlier and hopefully stop development of these complications. Which is why in 2011, the WHO recommended that hemoglobin A1C be used to diagnose diabetes. And they suggested a cut point of 48 millimoles per mole, or 6.5%, as a level above which the patient is diabetic. Going back to analytical aspects, if we look here, we know the level above 47 millimoles per mole, 6.4%, is diabetic. But then you've got low risk of being diabetic, a value less than 40 millimoles per mole, or 5.8%. That's only a 7 millimole difference, or 0.7% difference in NGSP terms, a very small amount. So our methods need to be as accurate as they can be so we don't misdiagnose these individuals. The same difference here. We can see imprecision. Our methods need to be precise, because if you see if a CV of more than 2% is found, we run the risk of actually misdiagnosing diabetes. Because if you got a CV of 4%, your method will overlap the diabetic range and those at low risk. So accuracy and imprecision are important. And clinically, we actually now recognize that we can be more or less stringent with patients. We should be more stringent if there is a risk of developing long-term complications. If the disease duration is low, we need to be newly diagnosed, need to be more stringent. Whereas somebody who had diabetes for some time, we can actually then fine-tune that treatment. If life expectancy is long, we need to be more stringent so we stop them developing long-term complications. If the individual is old, then we can relax slightly and give the patient slightly more freedom to actually have their life and enjoy it as they should do. So we now know that we should actually be treating individuals with the diabetes control. Talk about impact. Well, we have all suffered from COVID-19 over the last 18 months, but patients with diabetes have suffered even more. We know that older people had an increased mortality with COVID-19. But when we look at people with diabetes, we can see that at every age range, if you were diabetic, no matter what kind of diabetes you had an increased risk of death if you had COVID-19 than patients who didn't have diabetes. If you look at this All England study here at the odds ratio for in-hospital deaths from people with diabetes with people with COVID-19, we can see here that there's increased risk of death of all forms of diabetes. But why is this? If we look at this in slightly more detail, we can see that as hemoglobin A1C increased in type 1 diabetes, there was increasing risk of death in these individuals. Now with type 2 diabetes, we see the same again. As hemoglobin A1C levels increased, then the risk of death also increased. Hyperglycemia is closely related to increased mortality in patients with diabetes. Hemoglobin A1C again has shown itself to be an important marker so we can look after our patients carefully, in this case, patients with COVID. So the values we produce hemoglobin A1C in the laboratory are important. So let's look at the quality aspects. How well are we doing with measurement of hemoglobin A1C? Several years ago now, the IFCC CUPD, which is a committee within the IFCC, set up a Euro A1C trial where we looked at hemoglobin A1C results produced across 17 countries within Europe using 24 manufacturer methods. In 2018, this extended to 17 European laboratories and 6 other laboratories worldwide. And we looked at how well results have been produced based on sigma metrics, i.e. bias and imprecision. And we can look at this as the overall position, how individual countries are performing, how manufacturers are performing, and trends, how things change over time. Overall first. So within these European laboratories and laboratories further afield, we can see that performance is actually not too bad. There is very small bias in results here. What we should be aiming for, of course, is the bottom left-hand corner here, where little or no bias and no imprecision. What we've seen overall here in this trial is good bias. There's a very small bias difference here. What we're seeing is poor, but still acceptable, imprecision. We look per country, and we can see different countries perform differently. Overall, most countries perform well. They should lie within this dark line here on the far outer level of this graph. Most laboratories perform well. There are some outside. But you can see again, accuracy is quite good with little or no bias by most laboratories. Overall, about a 1%, a 1 millimole per mole bias within the different countries, which is very good. But I must say, imprecision needs to be improved. We look at different manufacturers. You can see here different manufacturers using different method principles. You can see some manufacturers obviously perform a lot better than others. And ideally, we need to identify these poor performers and move them out of the clinical laboratory and adopt better performers. And look at trends over time. You can see over the 3 years, now it's been 4 years, we've been doing this study. You can see there's been very little improvement in terms of imprecision, but the accuracy has got better. Accuracy will lead long-term to improvements in imprecision. You can see that some countries are improving, probably because they're replacing poor performance. You can see here between 2016, 2017, 2018, there's been continual improvement in the performance in Turkey. And now they're almost beyond the black line here, showing that the performance is acceptable. We can use the same kind of sigma metrics. Look at the CAP survey we looked at earlier. We can compare 2014 and 2018. So if you look at the sigma metrics, we can see here again the 2 sigma line is what you're aiming for, and that the large number of results are lying outside this dark line. That was in 2014. We move swiftly forward to 2018, and we can see here there's been a marked improvement. You can see here from the star that the overall mean in 2014 was way outside the black line you're aiming for. But by 2018, a star is just crept in the bottom right-hand corner. And how has this been happening? Well, we can see it from we can't directly see, but overall, we have lost poor performing methods and actually replaced them with better performing methods. And this is the way forward. We need to say to manufacturers, you either improve the method or we move to something different. So where are we? I look at this, the evolution of hemoglobin A1C in clinical practice. We had the discovery of a new hemoglobin fraction. The recognition of this new fraction in clinical medicine. The large groundbreaking trials of the DCCT in type 1 diabetics and the UK PDS in patients with type 2 diabetes. Standardization of results to the IFCC calibration. And these standardizations have allowed us now to set targets for our patients with diabetes, both for type 1 diabetes and for type 2 diabetes. And standardization has now allowed us also to use hemoglobin A1C for diagnosis. And hopefully with this tool, we can start diagnosing diabetics far earlier in the disease process, and we can hopefully stop long-term complications. And we also now recognize that global targets are not right for everybody, and that recognition of individual variation is such that we now have individual treatment goals. And we must look at this as being the way forward. We look at individual patients, we empower them to look after the disease, and we should recognize that in what we're doing and set individual treatment goals for individual patients. Thank you.
you, Professor John, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, great to see the big improvement in bias over the years for the HbA1c assays. What do you think is required for there to be as big an improvement in the precision between methods?
Thank you for this question. I think that the bias will drive improvement in precision in the long term, but it's really down to manufacturers now to look at their methods and actually look at what they can do to improve their own individual imprecisions, especially when it comes to batch-to-batch variation in reagents. Also, is down to individual laboratories to look at their within IQC and how they control their systems. So it will be, I think, in the future, a joint approach from laboratories, but I think mainly from manufacturers to look at their own methods and what they can do to improve that imprecision. But calibration with a true standard will help them to achieve this goal.
Thank you. Next question. I am still confused by the terms harmonization and standardization. Can you explain this once more?
Yes, certainly. Harmonization is something which is not unusual in laboratory medicine. It is when we don't have a true standard, we assign a value to a material, sometimes an arbitrary number, and then we use that to calibrate our systems so that you bring the results to a target. Usually that target isn't the true value because we do not have a means to assess that true value. Standardization is when we have a true calibration material, a true standard to which we can actually set our target goal to, and then we can bring all results to that true value. That gives us the accurate result for the thing we want to measure. So harmonization is bringing results together to an arbitrary number, where standardization is bringing results to a true, accurate, correct result.
All right, thank you. Next question. Will the NGSP determination be changed to IFCC measurements in the future due to the interferent effect you mentioned?
Well, that is very much of a country by country decision, and also organizations within that country. Europe as a whole has mainly switched to IFCC reporting numbers. Manufacturers worldwide use the IFCC laboratory network to calibrate their equipment. But when standards are provided to manufacturers, they're given the IFCC or the SI units and also NGSP values, which are assigned by using the equation that relates the two systems. So the calibrants that the manufacturer use are primarily using SI units. NGSP units are calibrated back from that. Why would you want to calibrate back to a number that you know is wrong? Well, some people think that they have to relate this back to the DCCT study or the UK-PDS. Trials that happened many, many years ago and possibly have little relevance in modern day clinical practice. So will it change? I think it should. But again, that's down to individual countries and also individual scientific organizations within those countries.
Okay. Next question. Why do you think that less than half the people who have diabetes are diagnosed, and how can more people be assessed? Should the HbA1c testing be mandatory at an early age when treatments are available?
Yeah, it's unfortunate that the large number of individuals who have undiagnosed diabetes tend to be in the countries where they have little resources to actually try and identify this disease in Asia and Africa, etc. And of course, the incidence in those areas are actually quite high. Incidence of diabetes in Asia is one of the highest in the world. But of course, the problem or the issue with type 2 diabetes is that the symptoms develop very slowly, and that you can be quite a long way into the disease before symptoms actually arise. Screening, as you point out with hemoglobin A1c, may be a way forward in looking at the disease prevalence and identifying the disease early. But as I say, many of these individuals who are suffering from type 2 diabetes are in parts of the world where screening with hemoglobin A1c is probably difficult or unlikely to happen.
All right, thank you. It looks like we have time for one more question. What is the status of the use of individualized treatment based on the patient disease profile?
That's interesting, because this is something which in clinical medicine we have been trying to persuade our patients to do for a number of years in all different kinds of disease states. And in diabetes, we have been very successful in engaging individuals with diabetes to empower themselves to actually take on the disease and treat it themselves. So they have glucose monitoring devices. They have pens to inject their insulin, and they actually can look at how they feel, what the glucose is, and then fine tune their insulin regime for themselves. Now, that has helped us actually to enable people to actually control their glycemia far better. But we have to assess individual treatment regimes. As I said in the talk, that if you've got somebody who is newly diagnosed with a long time expected life expectancy with type 2 diabetes, then you think to yourself, they may suffer complications. Therefore, we need to try and get that glucose values down to a good maintained level. Whereas if somebody who's had diabetes for many years, who understands possibly their diabetes and what is right for them, and also possibly their lifespan is less than they're older and their lifespan is shorter, they may not have the time to develop the complications that arise from diabetes. So you can give them slightly less strict regimes to follow so that they can enjoy some of the things they may enjoy doing because they will not have the time to develop the complications. So you've got to really look at the patient in front of you in your clinic, assess their needs, assess what the complications may be for them in the long term, and then adjust that regime to actually treat them as an individual.
All right, thank you. Well, thank you, Professor John. Do you have any final comments for our audience?
I'll just thank you all for listening. I hope you found the presentation of interest. It is very important, and I think that the message we need here is that you need to understand standardization, because until you understand the standardization of hemoglobin A1C, it's difficult for you in different countries to draw up guidelines for our patients and for clinicians and laboratories. And I will say that thank you very much for your interesting questions, some difficult, but I enjoyed answering them. And I'm sure that if you wish to ask more questions in the chat, then we will do our best to actually answer every question that comes to us. Thank you for your attention.
Thank you again, Professor John, for your time today and your important research. We would also like to thank LabRoots and our sponsor Abbott for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.